When was the last time you got a paper cut? And how long did it take to heal? We all rely on our body's ability to repair damage to the skin, whether it's a tiny paper cut or a major injury. We can even take this amazing healing ability for granted. But for a variety of reasons, many patients suffer from chronic wounds which fail to heal, and this can be both painful and dangerous. In my research, I'm interested in studying the ways that different cell types contribute to wound healing so that we can better understand what has gone wrong for patients with chronic wounds and how we can fix it. When you sustain an injury, your skin enters a series of specific stages that will eventually result in full healing. The first stage is called inflammation, and this occurs over about the first 3 days after injury. You might have noticed this happening if a cut becomes red or maybe swollen after injury. What's happening is the wound sends out emergency signals to the whole body and then recruits a massive number of immune cells to the site of injury. When these immune cells, shown in purple, arrive, they fight off infection from the outside world, and they clear away debris in order to set the stage for future healing. It's very important, though, that inflammation stop when it's supposed to, and that you don't have too much or too little inflammation. A wound that experiences insufficient inflammation could fail to fight off infection, but a wound that experiences excessive inflammation could fail to proceed to the later, more reparative stages of healing and become these dangerous chronic wounds. I study this process using a mouse model because our skin is very similar since we're both mammals. I use a tool called a dermal biopsy punch, and this is quite similar to a tool your dermatologist might use at the doctor's office to remove a suspicious mole. I can use the dermal biopsy punch to induce a wound in mouse skin, and then follow along over the course of healing to see which cells are involved and what they're doing. I discovered during my PhD that a receptor called AXL is highly upregulated in the skin after injury. AXL is known to be involved in determining how immune cells can detect and then respond to dead cells in other parts of the body, but it had never been studied in skin wounds before. I found that AXL is upregulated 24 hours after injury, which is right during that inflammatory phase. As we might expect for this phase, AXL is expressed by some immune cells, which are the main players in inflammation. This was very exciting for us because it suggests that there could be an important signaling role for dead cells in the wound bed that hadn't been fully appreciated before. This is just one of the ways that scientists are still finding new aspects of the wound bed environment. And the clearer our picture of the healing wound becomes, the better able we are to help patients with chronic wounds to heal quickly and effectively.